everyone. My name is Butyaev. Yes, today I'm going to talk about a little bit more application part, not the biological part. So at the beginning, I want to start actually from 2001. Okay, I think I have it here. Whoa. So 2001, this is the first release of human genome, right? It was a game changer in the area. Actually, it appears to be a huge source of the data. Also, it created the huge potential for development in this area. Scientists started to develop a lot of data, tons of the data, which is supposed to be analyzed. So that was a strong demand on some tools to manipulate this data. And UCSC and Ensemble genome browsers were one of the pioneers in that areas, and they still remain one of the most reliable tools. However, technology were progressing, and nature of data, complexity of data were changing. For example, 2009, high-C technique appeared, and 3D structures seems to be important for understanding some regulatory mechanisms. But that was a kind of problem because since then we didn't change any paradigms in storage part. We didn't change any tools, tools to manipulate with the data, which is already 3D oriented. So 1 year ago, we tried to change something. So we were working in two different areas. The first one is trying to store and access the 3D genomic data. So the question was, is it possible to efficiently store and, more importantly, efficiently retrieve the 3D genomic data? That's why we created the 3DBG, which is three-dimension database genome. It's quite hard to understand, but it is. Also, we were working in the visualization, and we were playing a little bit with the data from 3DBG. We created the light-weighted, web-based 3D genome browser. If you have a laptop or something like this, you can go there and try it right now, and it's quite interesting. So I'm going to go a little bit deep. So if you look for some ideas how to deal with spatial data, you will probably find something like this. So organization of the data, is it index-based or structure or cluster-based? If you create the sophisticated data structures to improve the performance of the system, or you physically cluster the data inside your machine, and in this way you improve the performance. If you create the adaptive system, so the system that adapts storage structure based on the query size or the data. If you, if it data organization, if a system, while you uploading or deleting the data, change its storage representation. Usually it says online or offline, it doesn't matter. So a lot of people right now working in more index-based systems. So they try to create the sophisticated indexes, but we decided to step in a little different direction. So we tried to create the cluster-based system. So that means we were trying to cluster the data, which is probably will be queried together. On top of this, we create the adaptive index, and we chose the adaptivity just because we didn't want to waste the resources because of the nature of the data. So as you know, a genome could be very dense in some areas and very sparse in other areas. And we believe that in some future, maybe not now, thanks to the previous colleagues who were talking here, data, 3D genomic data will be appearing more frequently. That's why we created the online system. So by our knowledge, our 3DBG is the first cluster-based adaptive online storage system. So we go in a little bit even deeper. So in very, very depth, we were using the OC3 data structure. So for every node, we have exactly 8 children. And it's very, very frequent, very, very popular solution for graphics area. So even, for example, this auditorium could be split in exactly 8 cubes that are not overlapping, like, for example, here, to understand where am I, for example. But after this, I don't care about what is going on above, right? So I will split only the thing is the bottom one. So for example, I'm staying right here, for example. And that was an application, and we were using these techniques. And as you know, genome could be sparse, could be dense. That's why we were splitting some nodes. Depends on the weight, depends on the how dense is this area. So a little bit more general picture, and this is the whole system of 3DBG. So when the client requests some part of the area, system goes immediately to the OC3 and try to get all the nodes from the OC3 with pointers that point directly to the physical data, which is managed by horizontally scalable distributed file system. In simple words, we just split all our job among a lot of machines. And in every machine, we try to maintain the highly clustered data. So in this way, our client always get the response immediately. Well, plus minus. So we were trying to compare the 3DBG with some state-of-art solution. And probably you know the most frequent way to handle the spatial data is R3, so rectangular trees. And we chose a PostGIS because it provides some kind of index, which is very, very efficient, and it's very, very simple to use. So we were comparing these two systems in terms of latency, and of course we were using the same amount of data. It was about 1.5 gigabytes of data. Also, we were retrieving the fixed size part of the space, and we're recording latency and number of records returned. So as you probably understand, as you can understand, higher latency, the bigger latency, the less efficient the system. So in this case, 3DBG is much more efficient in terms of 3D genomic data. So after this, we decided, okay, we're going to create the 3D genome visualizer. But the question is, how much data should we provide? If you say something like, okay, let's go ahead and show the whole genome, well, people will be a little bit overwhelmed, right? So I don't know how much sense it gives to you, but for me, nothing. So we decided to scale it properly and go ahead with the cubes. But how big this cube should be? And let's say we have linear size. I'm talking about my model. The linear size of my model is about 2,500 units. It doesn't matter what is the unit now. So we decided to compare 100, 200, 300, and 400 size cube. And of course, we were just curious to compare with PostGIS solution. So as you can see, almost all the 3DBG solution are lower than PostGIS solution that we've seen before. But right now, for example, if you see on the 3DBG for 400 units, which is right here, and compare it with PostGIS 300 units, which is right here, it goes a little bit lower than the solution for PostGIS. and sometimes repeat the shape. So it's a significant actually improve for the data, this storage system. But after this, we think, okay, if we want to just fly over the space and upload the data immediately, we need some threshold for visualization. So how the maximum less disturbing time response from the server. So we decided that it will be 200 milliseconds. And as you can see, the 3dBg for 400 units going a little bit lower than this threshold, and after this going higher. But it doesn't matter, because we think that most of our data will be right here, and just maybe sometimes going a little bit further, but if something happens, we can always go to 300, right? So with help of 3dBg and JavaScript, we created the 3D Genome browser. Again, you can go ahead and look for and watch what this. So graphical representation is built on these web services, the 3D structure, which is all these 3D curves that you will see on the screen. Nucleotide sequence, SNPs, chip-seq data, single nucleotide 3D coordinates. Yes, but we're going a little bit further, and this is the general picture of 3D genome browser in cubic mode. Yes, mode, this is the way how the user interacts with the system, or sometimes what type or what shape of the data user want to visualize. So for example, the 3D genome browser shows some data in 3D, of course. So you can retrieve for particular part of the space a lot of data for different strands at the same time and compare it. And in terms of, I don't know how about you, but for me it was very, very useful. After this, we were playing a little bit with colors, and we were trying to map 3D structure and chip-seq data. So right here in colors, color mesh, you can see the transcription factors, and the color means it measures the interaction. Also, you can see a lot of different links to external resources, which I will show you later. Also, we decided to create some kind of 2D genome browser to control. Okay, we didn't create it, we just built inside our genome browser. This is the genome maps, and it's already created. And with help of genome maps, we can control our genome browser, and we can move into the space with the help. Also, it contains a lot of information, a lot of useful information that you can use for research. So 3D genome browser also have a lot of useful features like gene SNPs mapping. We have back references to external resources like UCSC and NCBI. Also, which is pretty interesting, we can visualize the custom genotyping data. But because of everything is based on JavaScript, everything works on the user's machine, so nothing goes to the server. So the data is is securely stored on the machine of the user. We provide also the scripts for simple interaction with our system. So as I said, we built the 3dBg, which is a 3D-dimensional database genome, which is a storage system optimized for 3D genomic data. We created some web services for following investigation and so on. And 3dGB, which is 3D genome browser, which helps people to visualize genome, of course. You can find the genome browser here. Some scripting, some scripts for interact with the system. Okay, do I have some time? Because I can show actually the movie that I created. because here we don't have a so here we have our Genome browser, and you can go inside, and that's a linear mode, so we can extract the linear part of the genome. This is a cubic mode. Sorry, it's pretty fast. Here, 2 different strands, and now we trying to represent the ChIP-seq data. So as you can see, I'm moving. Yes, so click. Yeah, in here you can see all the ChIP-seq data, and we can go to UCSC Genome browser and find all the information about this range. So it's not so important. We're going further. This is some information about the different modes. We also provide all the instruction for people who are not familiar with this system. So this is the cube that I was talking about. So yeah, it looks like shooter, right? We uploading some data, and again, we want to represent the ChIP-seq data. And here goes a lot of data. Just a second. And there is a effect level, so we can interact with this. Right. Okay, so this is a Star Trek, almost. Star Wars, no? Right. So we are flying. We can see the 3D structure. Okay, a little bit skip. So also we can represent the distance between different parts of the genome, because it's very usually it's very important. So right here, for example, this is my current position in which model, and you can tweet if you want, but it's not so important. Yes, and let's say I want to see the distance between this point right here. Where? Right here. Yes, and on the right you can see that number 1 appear. Here I want to compare with this point. So the distance between label 255 units in terms of the model, of course. So now I want to compare this point and this point. And here 87 units. Right, so that's it. And I'm going back to, oh my God, how many? Right here, and I believe it's something like that. So I want to say thank you to ISCB for Travel Award. To come here was a great pleasure. And I want to thank my supervisors, Dr. Waldes Pohl and Dr. Blanchette, and their lab members. Also, I want to thank our Swiss collaborators, Dr. Kudro Maro and Ruslan Mavlutov. And thank you.